There we go. All right. Well, welcome everybody. Good evening. My name is Connie Sorman. I am an individual giving officer at the Children's Tumor Foundation. And this is our May NF2 meetup. We are delighted to have a topic that has generated a lot of interest tonight. So thank you all for being here. I just want to go through some housekeeping before we get started. Closed captions are available. We have a live captioner. You can click on the CC button at the bottom of your Zoom screen. Please mute your microphone unless you're speaking so that everyone can hear the presenters. We welcome everyone to introduce themselves in the chat. Tell us where you're from and what's your NF connection. And we will have a Q&A at the end of the presentation, but you can put your questions in the chat anytime and we will do our best to get to all the questions. And then during the Q&A, you can either raise your hand or if you have a question, you can put it in the chat. So first of all, I'd like to introduce Dr. Oliver Adunka. Dr. Adunka is the William H. Saunders MD Endowed Professor and the Vice Chair for Clinical Operations in the Department. Say it. Thank you. That's a difficult word.
ENT. Do what we do. Just call it ENT.
Head and neck surgeon at the Ohio State University. He also serves as the director of the Division of Otology, Neurotology, and Cranial-Based Surgery within the department. Further, he is the director of pediatric otology and the hearing program at Nationwide Children's Hospital, one of the largest institutions of its kind in the United States. Dr. Adunka has been the medical chair of the NF2 Accelerator since 2021 and has served as a member of the CTF-CCAB since 2016.
Yeah, thank you for this introduction and thanks all for joining. Yeah, it's a distinct pleasure to have Dr. Scott Plotkin with us today. Dr. Plotkin will talk to us about his experience with in the progress update on the INTUITT-NF2 clinical trial. Dr. Plotkin is a professor at Massachusetts General Hospital and at Harvard Medical School. He's the director of cancer neurology at Mass General Hospital. More importantly, for what we care about here mostly is that Dr. Plotkin is the chair of the CTF's Clinical Advisory Board. Scott has extensive experience with clinical trials, especially with NF1, NF2, and schwannomatosis. Scott has served as the principal investigator of many of these single and multi-center clinical trials. And he's an active member, very active member, I would argue, on several national consortia, including, again, the CTF here. He co-founded the Response Evaluation of Neurofibromatosis and schwannomatosis international collaboration as one of these examples. So obviously, Scott is one of the premier, if not the premier expert worldwide on NF2, and as a neuro-oncologist, we really welcome him today. Annette, should we, Connie, should we do the next introduction, or should we just have Scott speak and then?
Yeah, go ahead and do all of the speaker introductions now, and then we'll have them done. Thank you.
Okay, so then we have Annette. How you
Backer, right? Doesn't matter,
right?
as long as the kid has a name.
I'm originally from Austria, so German, and you're, I think, from Belgium originally, right? You're Flemish, right. So and I think Annette is a PhD and the president of the Children's Tumor Foundation, and Annette. You will talk to us about schwannomatosis platform trials and network the CTF has been able to set up to accommodate these clinical trials. So it's a fascinating talk, and there's a lot of work that went into that. So we can't wait to hear your talk, Annette. Thank you for joining.
Thanks.
And then Liana Scaglione, I hope I said that right. I actually spoke Italian a while back in my previous life, but so Annette, Liana is a patient with NF2. She lives in New York City with her 2 cats. And she was diagnosed when you were 16, I think, via a tumor in your lower spine. You found joy running, and recently I heard you've joined the NF endurance team. So you have participated in the New York City half marathon this past March, and you will run them. You plan to run them again in November for the full marathon. So good luck for that. This is Leanna's 3rd drug trial for NF2. And you've been really good at documenting every step of the way. So you can find her on social media, including TikTok and Instagram. So thanks again all for joining. Can't wait to hear the updates and what's new in the world of NF2 and schwannomatosis. And I think Scott will kick us off here, right?
Is that right? Did you want to go first, Annette, or you want me to go? Okay. Well, mine is really built on yours, but let's give it a shot.
I think, Scott,
would you prefer to revert it?
Well, let's
more logic. You're right.
Well, the only reason I would propose that is for anybody on the call who's not familiar with platforms. When I saw your slides,
Yep.
I kind of backed mine out so you and I wouldn't duplicate.
Perfect. So I'll happy to start.
But before we start, I just want to make sure that the captioning is at the right pace before we all start. So can somebody on the call confirm that captioning is working for everyone?
it seems to be working okay. So.
Yeah, it works.
Okay. I'll go after you, Annette, I think, please.
Okay, perfect. So good evening, everybody. It's a very exciting moment, I think, tonight. As Dr. Duncan said, first of all, thanks, Dr. Duncan, for the introduction. There's a lot happening in the NF2-related schwannomatosis field. So I think it's an absolute pleasure to show you a little bit what we have been doing. So what I am tasked with, and let me share my screen, is to talk a bit about platform trials. A lot of people are talking about it because it is very exciting, very efficient. But what are those, right? So first of all, this is in fact what was the start. Why are we doing what we're doing? Is what we see, what impedes clinical progress are 3 main things, and it's nobody's fault. It is just an observation, right? There are what we could say conflicting agendas, which means that for example, academia is really driven by publications, whereas pharma is more driven by keeping things secret, and you can go on and on. So let's call those conflicted agendas. The next thing, which is really important in rare disease, is that you need to have multiple sites that participate in a clinical trial, and very often different hospital systems have their own special IT systems, their special systems, and they're not always per se compatible. And then the third thing is that the investors are sometimes risk averse, and so it's sometimes hard to raise money to make everything happen. So I think I can easily skip this slide. This is about NF2 and schwannomatosis, so I'm going to skip this slide. But let me just generalize a little bit what is happening in rare disease. Why are trials in rare disease complicated? And there is in fact 2 main things. Neurofibromatosis and schwannomatosis are on the one hand a rare disease, which means that there is few patients and that you really need to do multi-site trials, which means that multiple sites need to participate in a clinical trial. But that leads to 2 main challenges. One, because of the few patients, it can lead to long recruitment times, and patient recruitment in a clinical trial is absolutely key. If you look at the statistics, approximately 1/4 of the trials in rare disease fail because the recruitment time is too long. So that's really a challenge that we need to overcome. The second thing is if you work with multiple sites, with every site having their own way of doing things, your negotiations to get this multi-site trials set up can really be long and cumbersome. The second challenge in a disorder like neurofibromatosis and in most rare diseases is that they're multi-organ, so that multiple organs are affected. And in NF, there's also progressive. So now here you're dealing with different manifestations, including different age groups. So you can imagine that this becomes a very complex protocol. What has CTF been doing? We have been trying to figure out how can we deal with those, what we call system problems. And I'm not going to read you this entire slide, but if you just read the main blue ones, we've been driving collaboration. We're of course fueling discovery because we're fundraising because of people like you. We're really accelerating innovation in collaboration with the field, of course, thereby sustaining hope, raising awareness, funding research, and advocating for care. As you know, our mission is very laser focused on driving research, expanding knowledge, and advancing care for the NF community. And our vision is obviously to end NF. This is a study that Dr. Plotkin, together with Dr. Blakely and Dr. Clapp have led. And that is in fact building a, what is called, let's now call this for the lack of a better word, a preclinical hub. And you don't need to read this entire slide, but you can see from the diagram that it was a pretty complex endeavor with about 12 groups for about 3 years. And that was to identify potential, what we call clinical trial ready candidates. Which kind of drugs would actually make it through this entire complex screen from both cells to animals to make sure that we're selecting the winners that can actually go into clinical trials. And out of this study, there were a couple of drugs that came out that were really effective, both in the cells and in the animals. What we have seen is that, okay, now we have done this once, now we are ready to build some kind of preclinical hub, which means that what we are trying to do, maybe first before I say what it is, what we're trying to do is, if a company has a drug, we want to be as quickly as possible to bring that drug from an idea to the clinic, and then also through the clinic to the patients, right? So that is why we're building now this preclinical hub, is to make sure that if we find a drug from a company, we can say, okay, we are that one-stop shop. We can test everything that needs to be tested, whether that is in cells, whether those are biomarkers, whether you want to test them in animal models, how can we deal with the data? PK/PD is a way to look at what the drug is doing to the body and what the body is doing to the drug. And we're also going to buy some drug libraries so that in fact we create that one-stop shop to quickly take drugs and select them to go into clinical trials for NF2. So now you can imagine that we're going to have this machine that is going to spit out, for the lack of a better word, at a very high speed, potential clinical trial candidates. So then, of course, what you need to do is you need to have an equally efficient clinical trial system to make sure that you can that you actually can take this clinical trial ready drugs and put them into the clinic. So what I think is really important is that in a traditional trial, you have every drug is one trial. So you have drug A, which is the blue, drug B is the orange, drug C is the green. So for every single drug, you have to design the independent clinical trial, recruit your patients, go through the trial, and then at the end of the trial, it either works or it doesn't. But you can imagine that this is potentially a sequential system, so things are taking a long time. Well, this is another way to design your clinical trials, which is called a platform basket trial. And that is exactly the trial that Dr. Plotkin will be talking about, where in fact, it's a unique way to quickly test multiple drugs in one setting. All these drugs are being tested in parallel. So what does that mean? You have in fact now 3 properties of a platform basket trial, which makes this so efficient. Number one, your patients are pre-recruited because your patients are with you. They're in a longitudinal natural history study, and the patients are followed over time and are in fact offered treatments as they go. The second property of these platform basket trials is that you have all the agreements with the different sites in place. And the third one is that you have that master protocol, which is kind of that overarching protocol where you look at what the drug should be doing. And Dr. Plotkin will talk later about what was exactly done in the INTUITT trial. But as you can see, these 3 components make these trials extremely efficient. I'm going to skip over this. I'm just going to say that Dr. Plotkin is going to talk about this very specific NF2 platform basket trial, and all these sites that I was talking about in the previous slide, those are 6 sites that also Dr. Plotkin will be talking about, and those are all pre-agreed. So now it's in fact a question of having companies come in and say, okay, we will plug our drug into this existing empire. So we have already 2, and of course we want way more. Where do we stand with this? So we have the master protocol is in place. All these partner agreements have been signed. There are 2 drugs that are in trial now, and we're really ready for expansion. And of course, we're now trying to secure the funding for what we call round 2. So what does that mean? The cost of such a trial is $2 million per trial. And those are what they call signal finding trials. And Dr. Plotkin will talk more about what they exactly are. But it's really to look for those drugs that actually work in a limited group of patients. There's about 40 patients per drug. And in 1 year, we know, or the trial takes 1 year, and then there is still some time, of course, for data analysis. But you see that this is an extremely efficient way to test drugs quickly. What is in there for the people that invest? We're now looking at what they call return on investment models. We're looking at if an investor would come in, what would he win if we would win? The second is this is a way to also get access to funds that are not per se only NF, because what we are building here, as you can imagine, is potentially expandable to other rare diseases. And then there is this ESG metric that many people are talking about. And that is, I would call that, we're trying to convince the companies to play with us while we are making them look good, right? Let's call ESG for a moment, the look good factor. So here we are, we've identified together with the drug selection committee of INTUITT, we have identified 5 drugs. So we're looking at about $10 million that we're in the midst of raising now. And it's a same thing, signal finding, 40 patients per drug or per drug treatment, gene therapy, anything, right? And then again, that 1 year trial drug. Why us? As you know, some of you know or may not know, but we have been pretty efficient and responsible in our spending. About 84% of our from 82 to 84%, it varies a little bit, is spent on research and medical, which is everything from research, medical, but also public education and patient support. So with that, I would like to end and say that I really look forward to ending NF together. And of course, we're reaching out to all the investors and with some contact information that in case they would be interested in participating and working with us, we are here. So with that, I would like to hand it over to Dr. Plotkin to specifically talk about the INTUITT trial.
Should we see if we have any questions so far? I didn't see any in the chat. Does anyone have a question? All right, well, let's get Dr. Plotkin up.
So Scott, right?
Yeah,
Yep.
should I share mine now?
Yep.
Absolutely.
Okay, you'll tell me when you can see it, please.
We can see it.
We can,
Okay.
but not in the presentation mode yet. Okay, now.
Yeah.
Is
Perfect.
it now
You
it's
good?
okay?
Yep. You
Okay,
good?
great.
Perfect.
Well, thank you very much for the opportunity to be here. Thank you, Dr. Adonko, for the nice introduction. Overly kind, I'll argue, and I'm sure many people on this call would agree it was overly kind. It's so nice to see so many familiar faces. Thank you to everybody for showing up. And I know that many of the people on this call have participated in this or another trial that has been run. And we thank you for your participation because this is a team effort. We know it is, and it takes people participating and doctors running trials to make this work. So thank you for that. I'd like to start picking up where Dr. Bakker left off, honing down onto the current trial. And what I was tasked to do was to speak about the second arm of the trial. Last year, about 1 year ago, we reported our interim results for the first sub-study using the drug called Brigatinib. And you'll see one image tonight of Brigatinib. The update there that you should know is that we have completed our enrollment for the Brigatinib study, and hope to analyze that data soon and present it. None of us know it yet, so it's in process. And of course, we're going to try to get that information out as quickly as we can, just as Dr. Bakker said. So these are my disclosures, which we always put on any presentation. And so where I want to pick up is a reframing of the issues that Dr. Bakker brought up in terms of what are the challenges for NF2 related schwannomatosis clinical trials. Here are some of them as I see them. First of all, until 2020, there was no available trial for most tumor types affecting persons with NF2. There was no trials for meningiomas or ependymomas or non-vestibular schwannomas. And this presents an opportunity for us to change designs so that we can enroll persons who have any type of tumor. In addition, we know that even if you're looking at vestibular schwannoma in a trial, you could be collecting data on other tumor types, and that was not routinely done. But of course, it's an opportunity when any person's being treated to look at all the tumor types that that person might have. One of the things that's so challenging is we realize that the delay between trials is way too long. So we have put into place designs that are going to minimize that regulatory burden. Then it's difficult to compare results across trials. Somebody logically says, what works better, everolimus or rapamycin or brigatinib or Avastin? What works the best? Well, it's been very hard to compare across trials. And so you're going to see this design will allow us to do that a little better than we've done before. And to the point before about companies investing in this space, we wanted to create efficiencies that would reduce risk for these companies to entice them to invest in our clinical trials. And then finally, we're always looking for promising drugs. And I think Dr. Bakker already touched on that we have 5 that we think would be good options that we're already looking at. That's really based on the investment that CTF and other groups like the Department of Defense have put into this basic research to find new potential drugs. So here's how I think about the design of the INTUITT-NF2 trial. It's what we call, and these are all key words here, an adaptive platform basket trial for NF2. And I'll go into those, but the rationale is to rapidly and efficiently screen therapies for a biological effect. And for our intents and purposes, we've defined that as tumor shrinkage, which is the term we use, radiographic response. We could choose something else. For example, we could be looking at hearing, but when we look across all the tumors that occur in the setting of NF2, the one thing that we could measure in all of them was tumor shrinkage with treatment. And this trial gives us the ability to look across multiple tumor types and then also to focus on individual tumor types. And our goal, like everyone's goal, is to move forward with confirmatory studies that allow us to get FDA approval so that these drugs are available for persons with NF2 and not just for persons with cancer or other drugs or other issues. So here's visually how we think about it. You'll notice that there are 2 gray boxes. The top gray box is what we refer to as a master study, and I'm going to explain what that means in a second. At the bottom is a gray box called individual drug sub-studies. So when we think about the efficiencies of a platform basket trial, if you think about a person who has NF2, who has a tumor that's growing, the platform trial concept is that if you create an infrastructure for all of those patients, regardless of whether the growing tumor is a vestibular schwannoma or a meningioma or an ependymoma, if we're enrolling the same type of persons, we can have any drug we can use any drug to study that group of patients and they'll be comparable. So you can see here in a platform trial, every person will have common eligibility. So they have to be a certain age, with certain kidney function, with certain other features. And we measure the response of the tumors using common endpoints. So we're using the same ruler, so to speak, to say, does drug A work and does drug B work? That's what a platform is. A basket is when you have these different tumor types. So you can see here that if the tumor is a vestibular schwannoma, that individual would be in the vestibular schwannoma basket, and likewise for these other 3 baskets. So this basket trial has 4 baskets in it. And the reason you can do that is we know that all these tumors are driven by the loss of the NF2 gene. So they have a common biology. So a person who enrolls gets randomized, which means that they get allocated to one of the treatment arms. And the point I wanted to make is for the master study, there are no medicines in here. This is simply an allocation procedure. It brings patients into a funnel, collects everybody in the same group, and then allocates them to treatment either with drug A, drug B, and drug C. And we mentioned that drug A has completed accrual in, I would call it almost record time, 40 participants basically in less than 2 years. Drug B is just launching. We'll talk about that today. We don't yet have a formal drug C, but I've already mentioned, Dr. Bacher's already mentioned that we have at least 5 candidates that we would like to move into that drug C. So if you're a person treated with Brigatinib, we're looking at a target tumor. That's the tumor that we agree is the one that's growing and should be studied. If that tumor grows, what happens is that participant has to discontinue that medicine because it's not working. But that individual remains on the master study. And when a new medicine becomes available, they are eligible to be treated. And so we hope that this will ultimately, when we're fully populated, have a very quick progression for an individual who has a growing tumor to participate in study 1, and then study 2, and then study 3. That is the vision of where we want to be. The challenge is we need to have enough medicines to make that work. And that's what we're aiming for. So here are the 6 sites that are participating. And I really want to acknowledge all the hard work of this group. And you can see that includes people at Mass General Hospital in Boston, at New York University, at Johns Hopkins, at the University of Miami, at Mayo Clinic, and at UCLA. And you can see here that we're trying to incorporate centers that have lots of experience with treatment of participants with NF2, but also to try to cover as much of the country as possible. And so for the master protocol to enroll, this study will enroll up to 80 participants. I already mentioned that there's no medicines on this. It's just a way to assign participants to a drug sub-study. We collect natural history, so we're checking in with people periodically to understand how they're doing. And you can see in order to qualify for INTUITT-NF2, you have to have a diagnosis of NF2-related schwannomatosis. You have to be at least 12 years of age. You have to have a target tumor that's growing. It has to be measurable by MRI. People can't be so sick that we worry that they won't survive, and they have to be able to care for themselves. We have some exclusion criteria that I won't go over, except to point out that all of us have tried incredibly hard to bring treatments to our younger patients. And you can see the age is 12 or greater. That represents a real monumental effort on the part of our community to ask these oncology companies to allow us to treat adolescents because these drugs are approved for adults and not kids. And so it's been really an important part of what we do and part of our commitment to the NF2 community. Now, if we look at when a person is enrolled on one of the sub-studies, you can see that what we look for. I already mentioned that tumor shrinkage is what we're looking for the primary outcome for us to determine whether the medicine is working. At the same time, we're looking for tumor shrinkage for the non-target tumor. So if you're in a study because of a growing meningioma, we also want to know what's happening to the vestibular schwannoma, what's happening to the spinal schwannoma, because that allows us to have more information about the medicine. We're looking for hearing improvement, and we're measuring quality of life. as well as safety and tolerability. I'm sorry that this is not on the next line, but we want to know all those endpoints. I just mentioned that Brigatinib was our first medicine to study. It is approved by the FDA and the European Medicines Agency, which means that if we get positive data, we're hoping that we can translate this into approved treatments for persons with NF2. And one of the nice things is this medicine is a once daily oral dosing. And while I use the term favorable side effect profile, that's probably because I'm a doctor who didn't take the medicine. I'm certain that some people on this call tonight would disagree with me, and I think they'd be right. But when we compare it to some of the other chemotherapy medicines that we've used in the past, this has been probably one of the single best tolerated medicines that I've worked with. And while it's not perfect, that has been a big plus. And the topic of tonight is Neratinib. This is the second medicine. It is a medicine that acts on a group of a group of proteins called the EGFR, HER2, and HER4. These are different proteins that are common in breast cancer, and that's why it's FDA approved for that treatment. Once again, it's a once daily medicine, which is very convenient, and the side effect profile is listed here. And I should say that we're in the process of learning from our participants, are there any specific side effects that are more common in NF2 than in breast cancer? And we're waiting to get that information. So this is how the study is currently going. Our goal is to enroll 20 participants. And the study requires that each basket, and remember there's 4 baskets, each basket is required to have at least 2 participants so that we can get data on each type of tumor. Because this is a drug approved for adults and has never been used in children, the FDA required us to do a safety run-in. What that means is our first 3 participants are being treated at 200 milligrams a day. We then analyze the data. If it's okay, the next 17 participants will have a dose increase to 240 milligrams a day. 3 plus 17 is 20. So we're going to do an analysis of the tumor shrinkage rate in these 20 individuals. We also have an associated study where we use PET scanning. This will be done in UCLA. And we're trying to see, can PET scanning help us predict who's likely to respond to the medicine and who's not. And then finally, after 20 participants have been enrolled, we're going to look how many people had tumor shrinkage of the target tumor. If there's 0, we're going to say that the medicine is not worthy of further study. But if there is at least 1 target tumor that responds and shrinks, we're going to look for additional funding for 20 more participants. The study opened in October. We've enrolled our 4th participant. And so we're collecting data still in this safety cohort. And then we hope to have that done very soon, within about 25 days or so. And with that, I'm happy to answer questions, but I do want to point out what a huge team that it takes to run these studies, and to thank everybody who's either participated or funded it or helped with some of the research. And so with that, I'll be very happy to answer any questions. If there are any.
Yeah, so far I don't see any in the chat, but feel free to speak up and ask your question if you have one.
I see one here. Let me just
Yep,
see if
here
I can
we
pull
go.
up.
Got it
Okay.
coming in.
Let me just try to get the thing. Okay, let me start with Maxine's. First question that I see is tumor shrinkage is the primary endpoint. How about stasis? I think stasis probably means tumor stability. And that is a great question. We have done extensive, one thing I couldn't talk about is we've done extensive interviews with some of our participants because we want to know, are we looking at the right endpoints? And I would agree what's come out of it is very clear that people who participate feel that having a stable tumor is a positive thing. It requires a different type of trial design to prove to the FDA that tumor stasis or tumor stability is real, that it's not an artifact or just a random occurrence. For example, they ask us to include participants who get treated with a placebo, which is a non-active formulation like a sugar pill. We've been very committed to not having placebos because we know that people do not want to enroll in studies in which they're treated with a placebo. So the way we're addressing this is we've collected information from MRI scans prior to treatment. So we're calculating a growth rate before treatment and a growth rate after treatment. And so we're going to be able to at least answer if a tumor was growing at 50% beforehand and only 20% after, so it's growing 30% less fast, we can present that data, but we're not yet sure how the FDA is going to receive it. So we're going to have the information and it's important, and we'll see what the FDA says about that. Second question, I hope that answered. If it didn't, put it in the text. Michelle Kroll asked, so are you still looking for I think participants? And the answer is absolutely. We've enrolled the first 4 of the 20 participants. The legal arrangements are in place for many of the other studies. I mentioned that we have to treat these first individuals for at least 28 days before we can present the company with safety data to move ahead with the enrollment of the final the second group. And so we anticipate doing that in June, that we should have that information in June so that we can start enrolling more broadly. But the other centers will be open for enrollment, I hope, this summer. And then from Mary, when will you share more results about how the Brigatinib is working? Where can we find the results that have been shared so far? Well, I think the Children's Tumor Foundation has a recording of that presentation, I believe, from January 2022, so last year, and I think that's available. And then I anticipate that we'll have some more information. My dream is by the Children's Tumor Foundation meeting in June 23rd of this year. I can't promise that because it has to, this information has to be seen by the company and so forth. So I don't want to promise that, but I can tell you that's what we're all working towards on our side. And then Michael says, is there a possibility of future studies that combine drugs with other drugs or other therapeutics, looking for a synergistic effect or other benefits? And he uses the example of Brigatinib plus bevacizumab, which is called Avastin, or Brigatinib in a physical intervention. And you read my mind. I've been very hopeful about an Avastin plus Brigatinib study, but I'll tell you something that's not shocking to any of us. Until we have the results of the Brigatinib study, the company won't entertain a combination until they think it looks good enough. So in addition to wanting to report our results just because we want to report them, we've also already started working on the possibility of that combination. I can't promise you what happened, but I can tell you we'll push for it very, very hard.
Yeah, and just secretly, it's one of the 5 drugs that we're looking for to look at combinations. So Michael, you got it spot on.
He wins the award. He's always been good. What kind of PET scan do you use? This is from Eric. What kind of PET scan do you use? MRI, CT. So PET scan is neither MRI or CT. Remember, PET scans, it stands for positron emission tomography. It uses a type of radioactive particle called a positron. And so typically the PET scan's done with a low dose CT. We pair them. They're called PET CTs. And it's a measure of how much glucose or sugar is taken up by the tumor. And we think for many kinds of cancer drugs out there, PET scans can help pre-select individuals who are likely or not likely to respond. Michelle says, can a patient be treated with Brigatinib if not on trial? The answer is yes. Technically, it's an FDA-approved drug in the United States, so a physician is allowed to prescribe it, but insurance is not required to cover it. So the battle is usually for insurance, which is another long story. Okay, David, would wanting my schwannoma in my right arm to shrink make me a participant? If it's growing, then you might be eligible, but if it's symptomatic but not growing, this study would not enroll you, only because we have to show that these medicines either cause it to shrink, which is reversing the growth, or cause stability. So we're only enrolling those that are growing. I see. Oliver is saying, Scott, hold up, we got more to go. So we should probably go to Liam, but should I answer any more or no? I guess that's it.
I think that's all that were there. You caught them all. So
Okay, sorry about that, Leanna.
It's okay.
so let's add
Okay.
Nina.
All right, you can switch it. I'm not gonna say anymore. I'll
Anything
be
else?
quiet.
Yeah,
No,
I'm trying
I
to
was
find
wondering, Connie, is Leanna supposed to share some of her,
She is. She's going to
experiences?
share some of her experience, but I think
Right.
it was supposed to be a conversation between
Yeah.
Dr. Plotkin and Leanna. So let me just
Okay.
get her on the screen too. Okay. There we go.
There you are.
Thank you for that. You ready?
Sure.
All right. So let's do this informal if we can. So I would say, would you like to say anything before we just start off with some questions, or should I just jump in?
No, you can just jump in.
Okay. Well, we know that, as Oliver said in his introduction, you're not a rookie. And what I mean by that is you've been a participant in other trials before. So which from my perspective as a physician trying to develop new treatments is so important. Can you tell us something about how you decided to be somebody who participated in trials, and maybe a little bit about your motivation?
So at the time when I was first introduced for the first trial, which happened to have been the Brigatinib trial way back when, my vestibular schwannomas were very, very slow growing, but they were still growing enough to still qualify me for the trial. And I also still had 100% hearing. So for me, I saw it as, if it doesn't work, there's no risk, but at the same time, I'm helping move research forward by at least providing an answer of whether or not it works for someone else, and possibly give hope and everything. And that's really what motivated me to continue on into my second trial onto the everolimus drug, which unfortunately I was not able to complete. I just did not have a good reaction to it. And I took a very long break, almost 10 years. And I am now in that group where the tumors are growing pretty rapidly, and being on a trial is a bit more personal of a motivation to finding an answer, but also at the same time, definitely driving to find an answer for everyone as well.
Thank you for that. That's wonderful. Let's start off. I have a few more questions about trials in general, but can you tell us a little bit about, tell me if you want to share this, about how the neratinib has agreed or not agreed with you? Because I'm sure a lot of people are wondering, what would it be like to take the medicine? And the only thing I'll say before you give us your answer is just to recognize that every person has different response to medicine. And while we'll listen to yours because you're the expert, I would say that there's always a little bit of caution listening to just one person, obviously. I'm sure you would agree with that.
Yes, and I also want to add a disclaimer to my reactions. I'm training for a marathon while I'm doing this, so I am pushing my body to its limits right now. So please take my reactions as kind of my body's just drained a lot of the time. I think the biggest one that I've really noticed is the fatigue. There are definitely the gastrointestinal issues that I noticed that were listed on that slide. For many times, I'm very lucky to have a work job and everything that allows me to be flexible and work from home, so I can have my bathroom nice and nearby. But the other crazy weird ones that I've been experiencing is food and smells are very weird. Coffee, for example, I can't drink it anymore. It smells really, really bad. It tastes really bad. Also food that is naturally bitter tasting, like spinach or a Brussels sprout, for example, it's extra bitter now. So it's not always as appealing to eat. Bland foods have kind of been my favorite, but I will say it's definitely taking a toll on the exhaustion level and the bounce back. So it's just a little on and off, like like I'll have a really good week and then I'll have a really bad week. But like I said, I'm also training for a marathon, so I'm pushing my body to no tomorrow with it.
Well, thank you for sharing that. Obviously, it's personal information, but we appreciate it. How much work have you missed because of this?
I think maybe 3 days total over the course of 6 months. And that was just because there were days where I was just so tired where I couldn't even tolerate lying reclined on my couch staring at a computer screen. I'd just rather be curled up in a ball sleeping for the day. But that was only 3 days. Otherwise, if it is a difficult day and I'm kind of, as I like to call, zombie mode, I'm still able to function. I'm still able to go about my day, still able to get my work done. I'm just moving a little bit slower.
Okay, thank you for that. And I think a lot when we write these trials, I think a lot about the burden of being on a clinical trial. And I'll say what I mean, but then I want you to expand because you're the person obviously who's had to grapple with it. So coming to clinic extra times, getting extra MRI scans, getting extra blood tests, all the things that we build into a clinical trial for safety purposes that are required by the FDA. Tell us a little bit about how you handle that or how burdensome, because we know this is I call it a hidden cost of a trial. That is all the extra time that somebody has to devote to this.
Well, I would say it's definitely a bit of a burden for travel for me, as I am in New York City and I visit you in Boston. So it's always an overnight stay. There's always a hotel that I have to juggle and figure out if there's rooms. My past visit, it was the Boston Marathon. So that was an extra burden. Go Boston. But it's a little tricky with timing as well, because while I have a supportive work environment and my boss is really supportive, part of me feels guilty for having to take the time off for this. But at the same time, my boss is just like, do what you need to do to take care of yourself, and we want you healthy. So.
Great, thanks. And maybe we'll open up in a second to some additional questions, but can you say some of the do you have any positives that have come out? How do you feel 6 months into this about your participation?
I say it's worth it. Honestly, right now, my results, it's not 100% what I was wishing for going in, but I'm definitely seeing that it's doing something. And for me, that's better than nothing. So all of these bad days or days off that I would have to take care of and just rest for a little bit extra, it's worth it.
Okay, thank you. Oliver, do you mind reading out, or could somebody read out some of the questions, just because it's a little bit hard for me to?
You're muted, Dr. Duncan.
Sorry, rookie mistake. I think MRIs are in Boston, right? Michael wrote that, right? So when you go to Boston, Leanne, then you will have the MRIs there, correct?
Yeah, that's every 3 months.
Mm-hmm. And I think Scott and I share some patients, that travel from Ohio, where I am, to Boston, and what I noticed too is the visits, but then it was sort of nice to coordinate with you, Scott, and just to collaborate. So from a physician to physician kind of collaboration, I felt that was really good. I assume you have someone in New York City who you collaborate with on your treatment, right? Yeah. Okay. So I think that's sort of one of the main things. How did you decide, Leanna, on clinical trial versus surgical management? As a surgeon, sort of, we're always, it's never really a straightforward decision, I would argue, right? And can you maybe elaborate a little bit there?
Well, for me, despite the size of my tumors, my hearing is really quite insignificant in hearing loss standards. There are musicians with worse hearing than I have in the world today. So that really weighed on that decision. And I knew removing them, I had more of a risk to lose a lot more hearing than I already did. And I wasn't ready for that yet. So I'd rather go for invasive, non-invasive options before surgery can even be an option.
And I think in general, I see you nodding, Scott. I think with good hearing, we usually, unless tumors are really small, where we can do what we call a middle fossa approach, you know, we would really always steer people away from surgery as long as hearing is really good, right? So I think it's a good point.
These are always hard decisions, you know, you make in
Yeah.
big teams with people in an ideal situation.
Right.
Let me see what else.
There's no, I don't have any other question in the chat here.
Someone asked if Leanna has NF2 or schwannomatosis, and she has NF2.
But to be fair, because this will come out
Yes,
very
I
soon
know.
from PTF,
I was thinking that.
just to preface this for everybody, we've gone through a renaming in 2022 where we're using this umbrella term of schwannomatosis for every genetic condition that predisposes people to get schwannomas. So NF2 is now going to be called NF2 related schwannomatosis, or is called NF2 related schwannomatosis. And so we would say in the most general term, currently, Leanna has schwannomatosis, or more accurately, NF2 related schwannomatosis. And we're still using our parentheses to say formerly called NF2, or formerly called neurofibromatosis 2. So.
And there's one other question, Leanna. When will you know what results you are getting?
Well, we get to discuss them every 3 months after my MRIs, but I think it's in end of October or middle of October that I'll get my final end results to see everything as in full overview of what's happened.
We'll
Okay.
keep going to, you know,
Yeah.
so.
And there is another question, sorry. Was Leanna enrolled in Brigatinib? And if not, why? And if yes, why did she stop?
I was not. I missed the enrollment period.
It went very fast. One of the real challenges is with 6 sites, we're trying to make sure we're geographically distributed. But if you do the math, when you open 20 slots in a stage, that's about 3 or 4 per site. And so it has, one of the things that we don't like about this design is that it puts a ton of pressure on everybody, the investigation team, all of our patients, to try to get into that small group. So if you have suggestions, if anyone has suggestions how to make that better, I'd love to hear them, but it has been very stressful for everybody.
Hey, Scott, I have a question. When you unenroll someone from the clinical trial and you stop the drug, you know, which one it is, do you see any kind of rebound growth happening? Or I think that's something that has come up with some of my patients, you know, as a question. So I was wondering if you can elaborate on that.
Yeah, it's a good question. I don't have the data yet, but we do have the data in the database because we are following people after they discontinue the study drug. So for example, if you come off of Brigatinib, people are still returning within about 6 months, and we get those MRIs as well. So we will be able to answer that. I will say that individuals who've come off of Brigatinib, some of them have gone on to other treatments such as bevacizumab or Avastin. So of course that makes it a little bit more cloudy. I would say we don't really have that answer yet, but we hope to have it. I've not seen personally huge changes in the tumor size coming off, but that's just my personal experience. And of course, with so many other participants, we should wait to see the real data, I think.
Yeah, thank you. That's really helpful. And I think there were some more questions. Mary asked, can you share what percentage of the people who started Brigatinib have come off of it?
If I had prepped with this question, I could have told you, but I didn't prep with it. So I'll tell you that our first person who participated started in June of 2020. And so we still have people on, so that's 3 years ago, and we still have our initial group of individuals being treated after 3 years. which is pretty amazing. Remember, because every tumor was growing, every person had a growing tumor to come in. So that means for 3 years without significant tumor growth. I don't know the exact number who've come off, but that is exactly the kind of information, because they're not all mine in Boston, just to be clear. So there's some people in California and other places. So my hope is to have that information in the next few months.
Great. And I think Aaron commented here that that will update the group with data once available. I guess that's I think as part of the clinical trial. Obviously, there's a you'll publish that, and I'm sure the CTF will help you and us disseminate these results. Yeah. So,
Absolutely.
yeah. Okay.
Leanna, are there other things that you'd want to convey to people? Is there anything else that you think is important? Because you're the expert, so to speak.
Trying to think.
Either that's been exciting about it or that's been hard. I mean, those are the two things. Or complicated.
Trials are definitely difficult. It does take a toll on you. It also, I would say, mentally takes a toll on you as well. You're in a very weak, vulnerable state. So with your weaker days do feel a bit more weak. But like I said, it is worth it, and it's just got to power through and just kind of realize that you're in good hands and you're being taken care of, and you can kind of stop whenever you're not feeling well.
That's for sure. Great. Okay. Well, thank you for sharing that.
I think one question that came up, Beth asked if how the data sharing works across all the sites.
That's a great question. That builds on what Dr. Bacher said. So we have a data sharing agreement among the sites. So all the sites are required to share their data, and we have goals that the data has to be in within X number of weeks after a participant is evaluated in clinic. And we've just finished cleaning that data, which is a term which just means making sure there's no errors in the data. So that's been a very important part of the sharing across sites.
And maybe one quick thing I wanted to add is that there is also a monthly meeting with all the INTUITT sites where there is a very open and constructive discussion. So I think that is also really, and that is all managed by Dr. Plotkin. So I think it's fantastic that you have this open conversation between the different sites.
And I think for the group in general, right, these clinical trials that Dr. Plotkin runs, and I run different clinical trials myself. I mean, these are very, I mean, there's so many kind of checks and balances built into these that we have to report to the FDA and other agencies, right? I mean, these are really very well structured, right? And obviously, Dr. Plotkin, as I mentioned earlier, I mean, he's the expert on these clinical trials. So again, Scott, thanks for this talk and the expertise, and Annette for sharing the updates from the kind of framework that the CTF is able to provide. You know, this is really remarkable. And obviously, Leanna, thank you for your story. Good luck for your full marathon, right? Good luck with the training, you know, keep the energy up. I mean, I think you had a lot of positive comments here in the chat, and we're all rooting for you. So
yes, thank you. Thank you, everyone. I just have a few closing announcements before we go. Special thanks for many of you on the call today who have helped to fund our NF2 Accelerator project and the INTUITT trial. And extra special thanks to Tracy Galloway, R.B. Harrison, the Toms family, the King Bodwin Foundation of Canada, Mark Reese, Carol Harrison Gallagher, and the Morris family. I wanted to let you know that we do have a strategic plan webinar on May 8th at 1 p.m. That will be, I think Amy's going to post a link in the chat for that. So please join us for that. Also, May is NF Awareness Month, as you've probably heard many times already, but we have a great May 17 live event happening on World NF Awareness Day. We will be debuting CTF's new 2-part short documentary following 2 families, one with NF1 and one with NF2. And also highlighted our mission moments and NF hero stories from our Make NF Visible video competition. So be sure to sign up for that and tune in. It's going to be a great night. You can go to ctf.org/live for that. Don't miss it. In addition, we talked a little bit about the NF conference coming up in June. In addition, the NF Summit will be in June. So they're back-to-back this year. In Scottsdale, Arizona, from June 21st to the 27th. We hope you can make it to those as well. We do have virtual registration for both events. So nfsummit.org and nfconference.org are those links. And if you have any topic suggestions for upcoming events, please email us at nf2@ctf.org. Thank you all so much for being here tonight, and have a great rest of your evening.
Thank you, Connie.
Thank you. Thank
Good
you.
night, everybody.
Thanks,
Thank you
everybody.
so
Good
much,
night.
everybody. Thank you, Liana, Oliver, Scott. Thanks.